study guide for those of you who check your message, and you should always, as you go forward in life, check every message. Yeah. You don't have to respond to everything, but check them because it's people. People often make mistakes and they kind of, oh, I didn't see that. You have to learn to see that. Just a mini lesson there. So because of that, I sent you the study guide. If I'm feeling ambitious, I will try to do the review recording because I pretty much have finished modifying and playing around with the exam. I'm thinking either the 23rd or the 25th, which would be 2 weeks from, because we're going to finish this bacteria in the eukaryote chapter on the eukaryotic microbes. Doesn't take very long, and then we do the basic physiology, like how they grow, and really there's just several sections of that about how they, what kind of, do they need air or not, temperature levels, something called the bacterial growth curve, and that doesn't take as long as you think. So I suspect no later than, I would dare say, the 16th, either the end of next week or the 16th, we'll be done with this first unit stuff. So I'm thinking that gives you a full week for after we're completed with that, which I usually do. And so I haven't figured it out yet which one's going to work. I don't like to do it on Friday because, one, we don't see as many people on Fridays, and two, we have a lot of athletes and things who travel, and they're usually gone by Friday. Okay, so now you'll notice I added something else. I added this, and it's a zip drive, basically a zip file. Hopefully you can unzip it. If not, you could. I mean, you're gonna see it on the video, but you can. And there's several of these as we go forward in this and subsequent units where these animations are very useful. So we'll see one today on the endospore. The endospore is a big piece, the last probably big piece of this initial chapter that's coming through. So and so hopefully, if not, you'll be able to see it. What I did when I downloaded this, showed it, was in my file, this animation zip. I mean, we have a lot of things, but the one I'm talking about was this, and nobody has that shockwave player anymore. This is MP4, and it's like a legacy MP4. I don't know. You may know better than I, but if they ask you to open it up, at least our system here at school, if you're on a school computer, will be able to help that. I am very limited technologically, even though better than most for someone who's born in the pre-technology age. But I digress. Okay, we got all that introductory stuff. Good. Let's get started. So did I start this? Did I get this party started yet? I never know. I always forget. Yeah, I did. All right. So let's go. Okay, where we left off. So, I mean, the endospore is the big topic today. That's a major component. I'll jump around to a couple of the PowerPoint we were using, show you and complete it a little bit with the other PowerPoint that we were using, both saying chapter 3. I had gotten to this in the other class, but I went back and I reviewed it today because it's really important. And this has to do with the genetic material. I don't want to show my hidden icons. I just want to see the darn slideshow. Thank you. Moving on. So one of the major distinctions, and this really becomes important when we get into the eukaryotic cell, is what's called the bacterial chromosome. There's no nucleus. We call it the nucleoid. That's where it is contained in a structure that it's basically our DNA, eukaryote DNA. It's both double-stranded DNA. The DNA is the same, A's, T's, GC's, that whole thing. We're not going to get into it's double. When we get to viruses, things get a little funky there. But linear pairs, we all have, you and I all have, and all eukaryotes have a certain number of pairs of chromosomes because they're generated. Those cells are generated by mitosis or meiosis, and so we've got effectively you got one from the male parent, one from the female parent, assuming that was the typical kind of reproduction that was involved, though it's not always the case. And the beauty of that particular system, it's genetically friendly. So if there's a mutation or an error on one strand, only one strand is expressed. And sometimes you get, and there's a whole lot of genetic safeguards. And this is sort of laying the framework for why our cells are so much more genetically stable and less likely to mutate than bacteria. It's very easy to mutate bacteria, and they already are genetically unstable to start with. And we'll go into that when we do the genetics unit. So this is really an important distinction. So you have this giant circle, and it's supercoils. It's like when you were a kid, supercoiling, and you may have heard me tell this before. I mean, that's kind of what happens to our DNA when it's ready to replicate. When we start mitosis and you get in prophase, what happens is the chromosomes supercoil. They're like tiny strands because those histone proteins are holding them like that. And when we get ready to undergo mitosis and prophase, they start to supercoil. It's like that. You had a kid, you had that big rubber band in a plane, and you with a propeller, and you kept twisting and twisting, and it began to kink up. That's supercoiling. These are so supercoiled, this giant circle of DNA, that when a bacteria dies, it explodes out. It could wrap around that bacteria. Are you ready? The individual cell 1,000 times. Like you and I have about 35 to 50,000 genes on our DNA, all our chromosomes. Bacteria, think of the size comparison, have about 10,000, 5 to 10,000. And they're surely a whole lot smaller than 1/10 of our size, let's say, arbitrarily. So there's a lot of genetic information and a lot of bacteria. Viruses are a whole different subset of things happening. We have a whole unit on those that are there. So we call it the nucleoid, this place where they're sort of aggregated. And then there's cytoplasm and there's ribosomes. Not a whole lot going on in that cell, the bacterial cell. Most of them have plasmids. They don't have to have them because they're non-essential. They're tiny circles of DNA. They are the framework that we use not only for how resistance spreads, and we have a whole unit, again, with genetics that we go into it. They're tiny circles with maybe 20 to 50 genetic markers, and they are what we use for genetic engineering. We tend to use E. coli, and if we're doing eukaryotic engineering, we use yeast, because there are strains of E. coli and these are both so easy to grow, and that's why we use them, that are very receptive to accepting, let's say, foreign or different genetic information and incorporating it. We'll talk about that when we do the genetic unit. And that's coming in the next unit. So I'm trying to lay the framework. So what we do with these are tiny circles of DNA, and they have them over their plasmids. Non-essential means because they can come and go as needed. If it's like now, if it starts to snow, maybe there's an anti-snow protecting from the snow plasmid, just joking around. It'll start, and and all of a sudden in April, it goes away. It's very interesting how they have that capability. If only we could make it that fast. Why did I come back from Florida? And so drug resistance, protective traits, blah, 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 blah. We'll get into it. But remember, DNA gets moved that way. We splice it to the plasmid, and we use it in yeast and E. coli cells, particularly when we're moving it. The ribosomes are 70S, as you can see here, as compared to our 80S. It's not size so much as its density. Ribosomes have two, a small and a large subunit. They both have a very intricate structure made out of ribosomal RNA and proteins, and that's where we do protein synthesis. So, there's not a whole lot to it that we're not going to go into it. Even the archaeal ribosome is 70S. Next, when we get into eukarya and we start talking about the common ancestor and something called endosymbiotic theory, you're going to see this is a big player in that, because our mitochondria really resemble tiny bacterial cells. Very, very interesting. And that the difference between our cells, the typical eukaryotic ribosome at 80S and the bacterial one at 70S, it's a density. They don't add those pieces together. When they come together and combine, they have the S units are about density, okay, not about size per se. Let me just show you a picture, which is neat. And then everything else inside the bacteria that you may see in the lab is about ways that we identify it or things that are useful, food storage. So you're going to see these, we call them inclusion bodies sort of generically. gas vesicles, when I talked about things that could move toward the air or float, okay, they're useful for that. These iron oxide and different things are for magnetic properties. They can be either attracted or repelled by magnetic currents. They have micro compartments, so and different things that are involved with regard to it. By and large, in our kind of a lab, we use these for identification that are there. We used to not think that they had a cytoskeleton. Now we know that there's a cytoskeleton. It probably plays more in a role in whether it's going to be a cocci or a bacillus, or in ways where it reproduces to maintain its shape. It's not nearly as elaborate. Basically, they're very, very thin filaments like our actin or microfilaments, and they're right under the membrane, kind of reinforcing it. In our cells, those are more diverse. They do not have intermediate filaments, things make desmosomes, and they don't have microtubules, which is really significant. Microtubules are the things that make flagella and cilia. Even though they have bacteria flagella and eukaryotic flagella, they're totally different. Remember, and we'll get to this and I'll reinforce when we do the eukaryotic cell, flagella and cilia are inside the cell. They just push out, and then imagine this being, okay, they're sheathed in membrane. So they're within the cell. They're just making the cell appear to protrude. Where in bacteria, the flagella is at, and the pili and the fimbriae are actual, they're like hairs. It's like magically it did this. Okay. See that brilliant display? I spare no expense in giving you dramatic displays. Somebody could respond more favorably, or we're going to do this one. Thank you. I know it's Friday. I know it's snowing. I know you want to get the you know what out of here, but that's okay. We're stuck. I'm here. I got a faculty meeting at 3 on a Friday. Take my boss, please. So very, very important. We will look at it down the road. And this is the whole ball game, really the focal point for today, are endospores. And we'll have an animation that does, and that's why I loaded. So for our purposes, we really only see them in 2 genus or genera, as the appropriate plural, Bacillus and Clostridium. They're the ones I talked about when I started presenting these things about tetanus and soil, and effectively they're indestructible. The big thing is this: vegetative cell. Understand that where we develop the language, and I'm doing this because students miss this question all the time. A bacteria was thought to be a microscopic form of plant because that's cell wall. And so when bacteria were growing and doing normal activity and doubling and tripling and whatever, all the things that you saw in the lab with your plates filled with bacteria, right? From all those samples you took. That's called a vegetative cell. When a bacteria is, or a plant is vegetating, it's happy. It's replicating. Hi, what'd you do all day? I vegetated. Yeah. What a day. I know, but if we describe one of us, if you're on the clinical floor and say, oh, patient and such is in a vegetative state, what does that mean? They're like big trouble. They're very close to saying goodbye. I don't mean to be flip about that, but a permanent vegetative state is someone who effectively is either comatose or is being kept alive by machines, etc. Whatever, without getting into the ethics and the nuances that are associated with that. But so understand, when you see vegetative, and only these bacteria, only for our purposes, only bacilli and clostridia have this remarkable ability, depending on conditions, like all of a sudden it gets miserable out, and adverse conditions to them are not enough nutrients like carbon and nitrogen compounds and not enough moisture typically. Those are the things that are essential for them, nutrients and water, which all living things have to have. And they can magically go into a state of suspended animation called endospore formation. And to that end, let us look at this animation. If I didn't misplace it, which I probably did. I'm such a dweeb. You shouldn't let me do that. I'll get it. Where did I go? What a doofus. Uh-oh. Should be here. Oh, heavens. I got in here early. We don't have anybody here earlier on Fridays because they don't do the
spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions.
as late as loud as I can make it.
When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
Now that's the whole picture. Yeah, we hear you. I'll go over it again. First of all, they're both rod shaped. They're both gram positive. So really, for our purposes, those are the only. That's an important thing to note. The significant difference in why we utilize bacillus in the lab, it's an aerobe, so we don't have to have it under anaerobic conditions. Why we are so worried about tetanus and botulism and gas gangrene and C. diff, they have to exist in a special environment without oxygen. So when there's a nasty wound and it undergoes an interesting process, sad, called necrosis, necrosis, means the tissue starts to die. When it dies, it's generally from lack of blood supply, and it turns black. And what happens, that's a perfect breeding ground. That's what we call tetanus-prone wounds. I have a really good unit at the end on wounds. I have a tremendous amount of experience dealing with wounds. I've seen cases of tetanus. I've seen numerous cases of gas gangrene. It's not pleasant. Okay. So, I mean, to understand that there's a lot of relevance in understanding endospores. Again, this whole idea, what makes it so impervious to destruction, the definition of sterilization is the removal of all microbial life, including bacterial endospores. If you get them, everything else is gone. That we know, that we scientifically know is removed. You'll see prions are not yet in the definition. We don't know enough about them. And we'll get to that. We start talking subsequently in the next major unit that we do about sterilization and disinfection and things like that. But really, that's how we do it. If we get those, we don't have to worry about the E. coli. We don't have to worry about staphylococcus. We've got all the viruses, all that good stuff. Tuberculosis, see you later. So that's why it's so important to understand it. So what happens, without me going through it again, is just to show you why is this. Is I do this linearly.
Nutrients are depleted, or conditions
so
nutrients are depleted.
You see that normally bacteria just divide in two. But when conditions are adverse, as they're commenting, and I'll get rid of the sound. When conditions are adverse, what happens? It begins to form in this fashion. And it divides not symmetrically with equal amounts of cytoplasm, but asymmetrically. So on one side, as you will see, it forms what's called the mother cell on that side, and this is what's called the forespore, which is going to become the spore. So it has, even though they don't do it to scale, the DNA is the same. And this is why it is not a form of reproduction. It's a transition from vegetative to the endospore or spore state, called sporulation. And so you have, not depicted accurately, but you have just enough cytoplasm to be able to keep this sustained when it comes back to its vegetative state. When conditions change, once it has moisture and sufficient nutrients, it will start to grow. Just like if it could be tetanus or gas gangrene or botulism or whatever. Moving on. So what happens, the mother cell envelops and effectively, notice why it's not reproduction, the other DNA degrades. You don't have two new units. The DNA from one degrades. That leaves us, and it utilizes the membrane from the mother cell to create a double membrane, reinforcing it, if you will, allows peptidoglycan to form. They're all gram positives. And that forms something we call, as it firms up, called the cortex. And so now inside, interestingly enough, what happens is a chemical is produced that has kind of unique properties. One of them, it's called dipicolinic acid. It attracts calcium, hardening it, and it expels water, dehydrating it. When you preserve something, one of the easiest ways to preserve them is to dehydrate it. When we have a food dehydrator at home, we get certain stuff that will rehydrate. Okay, if we buy excess of, or my wife grows excess of, I mean, if you go out and buy, effectively it's dehydrated stuff that you buy. I mean, it's not really an exact example, but dried fruit would be like that. You know, if you like craisins or regular raisins, that it's not quite the same. I mean, if you go out and buy mushrooms in little bags that are there, that's because you want to get like a fancy mushroom, you rehydrate it, and you get the same effect. You do have porcinis because they're kind of hard to find, kind of expensive too. So all of this leads to that, and the dipicolinic acid does more than that. Now, once that's done, effectively, you've created this sort of unique entity, and now it forms a protein coat and even an additional layer of protein in certain cases. And that is really remarkably resistant to, I mean, extremes only of radiation, of heat, as you will see when we go into it. Allow us to remove those. We can't. You want to boil them forever? They're not going to go away. That's not sufficient. It isn't just easy. I mean, ultraviolet light. You've got to hit them sustained with what we call ionizing radiation. And I will go into all of this when we do that unit, which is coming up after we finish how they grow. And so that, I just want to share that with you so you had that. And so it's easily obtainable from here. I'll just try to get this link and put it in, and hopefully you'll be able to play it satisfactorily. Just rewatch what we're looking at here. And so there it is, induced by environmental conditions, extraordinarily resistant to heating, drying, free. I mean, you can't do much with it. Honest to goodness. I don't get a lot out of that, but if it helps you to look through it, go ahead. Talked about the different ones already early on that are there. Again, this is a classic example of a test question. The students, it's so easy. You know that they don't have membrane-bound organelles. Nucleus goes away. Nucleolus is only inside of a nucleus. It goes away. Nucleocapsid, you're going to see, has a lot to do with viruses. It doesn't have pertinence to bacteria. I mean, process of elimination, that's the only answer. Nucleoid. Looks like a nucleus, but it's not. That's what OID means. Not doing a lot on archaea. We don't have to really include it. Of interest, a lot of their ribosomal RNA and little DNA subsequences they've analyzed are closer to us. We're probably closer, genetically speaking, to archaea than bacteria. I don't know enough about it. I'm sure it's an interesting area for research, but I haven't really, and I'm not going into why. And I was telling one of my colleagues here, and she was explaining about how the membrane was composed, how it was able to resist some of these extremes. Very interesting. So, but it's not for us. And so there's not a whole lot else to say. Berge's manual, just to complete it, because it's mentioning, is what you use in lab. You do what's called the dichotomous key. You're going to have unknowns in lab, right? Didn't Dr. Dress tell you, wherever you have for it? Most of the time we solve unknowns. just start doing it right away. Is it a rod or is it a cocci? Is it gram positive or is it gram negative? Does it have spores or not? Does it ferment this sugar? Does it have motility? What does it look like on the plate? Does it have pigment? All of those, it's yes or no. That's what dichotomous key means. And basically, it's like it's like you're following it. Yeah, okay, I go there. No, I go there. Yes, I go there. No, I go there. That kind of thing. And and what we do in a bacteria, in what we call a bacteria lab in the hospital. In the hospital, when there's gunk coming out of something, you go, well, that looks like an infection. You get in there and you get it deeply into it. You get a swab. You put it on a plate just like you did in the lab. You put it on several plates, and you start to test it right away. And you want to find out, is it a bacteria or not? And if so, what antibiotics or what agents can we use to get rid of it? It's called culture and sensitivity. Culture is to identify it. Sensitivity is what can we use to treat it. So important. And it's basically what you're doing in the laboratory in a lot of ways. And you're going to see the tests, even though they do multiple tests, we're more limited because, as you'll see, the most expensive lab in any college or university at this level, typically undergraduate level, is micro. There's so much media that you use. So, and that's what Bergey's manual's been around forever. And I don't do any of that stuff. And I'm not really doing. When we start that E. coli I talked about, that 0157H7, that's when they're typing it. When we say type it, it means we're using these markers, surface markers, and being able to see if we can attach antibodies or identifying markers to them. That's just how it works in the lab. No big deal for us. It's not about lab, and that would be in an advanced micro lab. Now do that, and we're done with that. Oh, thank you. One down. One to get rid of and one more to work on. This is the end of this one I was talking about before. This is that previous one that you saw, all those little taxis and stuff like that. So that's what I mean by the nucleoid. All of that red is where the supercoiling occurs. If you go back, it'll say compaction of, some of it's very sophisticated on those animations on that file. If you watch how it compacts, supercoiling is very interesting. I'm not going to talk about it other than that. But the whole idea, it's circular but double-stranded plasmids, essential but useful, non-essential rather, but useful, and shared antibiotic resistance. We'll see into that. Here's the ribosomes. Importantly, medically, antibiotics impacting 7S ribosome do not affect 80S ribosome. The big problem is, wait till we get to our mitochondria, and I reinforce that we have them in there. The cytoskeleton, storage granules, gas vesicles for buoyancy, endospores. 100 years is very, very inaccurate. It's much, much, much longer. They've done carbon dating on these things. You'd be amazed that they're there, and they're literally everywhere. It's the only way and as again, it takes about 8 hours for them, roughly, probably as little as 2, and probably as much as 8 to 10 hours to form once the environmental conditions change. And effectively, this dehydrated state, if you want to think of it, is like that. And germination, which we can trigger it in the lab. We can cause them to germinate, just like you would a seed if you were growing it at home. And you have a garden at home, and you got a pack of seeds, whether it's flowers or stuff you can eat, which we do. Not a means of reproduction. I mean, that has to be emphasized. And now we're going to get out of that for now. And finally, the eukaryotic cell. I'm trying to get you guys caught up. I'm slightly behind for whatever. It's easier on Fridays because the calculus students only have Monday, Tuesday, Wednesday, Thursday. Yeah. I used to know all that stuff. I mean, I was a math major in my early career. Key to the universe. But if you did not know, I was a chemistry major, and I did it because I was so good at math. It's not like I wanted to be a chemist with my life, but if you're good in math, you can be good in chemistry. So these are eukaryote. So this is what they're talking about, this thing called the last common ancestor. It's part of this interesting thing, which is we call it's all one word, I just don't know. Symbiotic theory. Effectively, endo is inside, symbiosis is two different things getting along together and making a positive out of it. And so effectively, it's almost like one tiny bacteria swallowed another tiny bacteria, and rather than destroying it, it became part of it and it replicated along with it. And that's exactly what we will get to when we look more closely at it as we get going in these chapters. So our ancestral cells billions of years ago, and now what they're describing it is, as you can see, they italicized it here, the last common ancestor. There's some of that in this chapter we will look at. I think it's now what they're calling endosymbiotic theory. I'm not doing evolutionary microbiology here. I'm just giving you an idea of why it's so important to understand. That's an important thing that if you're moving forward. I told the other class, if you're over in SCAFE, and many of you are going to take a lot of classes over there, whatever your major is, they just went nuts. The folks who were here a year ago with their first pharmacology test. Maybe you heard about it, maybe you did. It was Wednesday. Whiteboards there are filled with a lot of the stuff that I'll be teaching you. All the antibiotics they had, and this was antibiotics, antivirals, antifungals, what this test was. They were going bananas. I do this clinically. Last term, I only had one section of micro, rather than having two this term. And so it's not a disadvantage for the people who don't have me, but it's maybe a little bit of it. And look, if they don't have me, and keep a hold of these videos, particularly with that when we get to antibiotics, because you could help them out. Moving on. So first primitive eukaryotes were likely single-celled and independent, and over time they changed. I'm not doing the history of that. Don't know enough about it. Specialization, complexity, I'll buy it. Okay, is evidence that mitochondria they have a circular chromosome. Okay, they have the 70S ribosome. Our mitochondria, if you should remember this, they have the remarkable ability. Like if you haven't been to the gym in a while and you start going, I want to work out, you're going to make more mitochondria for your muscles. And all of a sudden, I got an injury or I got busy and I can't go to the gym as much. All of a sudden, your muscles are not going to need them as much. There'll be a tiny amount of change or atrophy. And what will happen is you'll begin to lose those mitochondria because they won't replicate. Mitochondria don't last forever. They have a lifespan. And eventually, when they fulfill their useful period, they disappear. They're kind of gobbled up intracellularly. So they have independent division, and I suppose they can be inhibited by antibiotics, certainly, because we do have membrane antibiotics. They're not very useful, and all the choices are correct. So the big players are the mitochondria have a single circular chromosome, and they have 70S ribosomes. That's why antibiotics that target bacterial ribosomes hurt us, and we have side effects frequently because all our mitochondria have these. There's going to be some complications or adversities, might be a better word. It's going to affect it, and most of those happen to be gastrointestinal. Get into all that. So the flagella is different. You're going to see that in a second, almost immediately. And this is important. A lot of the questions that you will see on this exam are phrased in prokaryote or in a bacterial cell or in eukaryote. Read the question. If you see the term nucleus and the question's about bacteria, it's a wrong answer. There ain't one. Unless everything is true except. For those who haven't had me, talk to somebody who has. I get very annoying tests, right? Depends. PIA, I know. I know you're looking forward to it. Think of it as a challenge. There it is. 9 plus 2. Remember that? Okay. Again, that's what I was showing you before with my clever demonstration. Flagelli, and they're much, much larger, much, much longer. Again, this is a teeny tiny cell that we're thousands of times bigger than, maybe even more, disproportionately much larger. That's that famous 9 plus 2 pattern. That's part of evolution. In a protozoan, that's our earliest ancestor. Their flagella and their cilia have that. Microtubules, they're sheathed in membrane. Whereas in bacteria, it's just a repeating protein called flagellin. And again, it just protrudes like a hair. It's not an internal cellular appendage. It is an extracellular appendage. So they're totally different. If it says bacterial cilia, it's a wrong answer. We don't, they're non-exist. Okay. They have flagella. The smaller version is a pili, smaller still is a fimbriae. So that's important. And those are all made out of microtubule. Different than bacterial flagella, thicker, more complex, rather than just a line of proteins. You have that 9 plus 2 of microtubules that are there. Cellular membrane covered, long sheath cylinder, regularly spaced microtubules. Cilia similar but smaller, more of them. And the reason they're important because we classify protozoans, how they move. Do they have flagella? Do they have cilia? Do they move by what's called amoeboid motion? They just kind of scooch around. Or do they not move at all? So it's all about classification. That's stuff that you'll see a bit in the lab as well. And again, they are clinically relevant because they cause disease. My kids had a protozoan disease that was very, very messy, and it came from contaminated well water when they were very little. Because there was, well water can get contaminated, unfortunately, somewhat easily. They all had giardiasis. That's a protozoan. That ain't fun. Thankfully, we have treatment for it, even back then. So they don't remember it. Plus, just like us, they have a glycocalyx. You remember studying the eukaryotic cell? Many of you with me? Okay. Hold on. Got it covered. So, we talked about before in AP1, all of our cells have sugars on the outside, some for identification, some to attach proteins, some to attach fibrous structures, And so, just like bacterial cells do, it can be things for attachment. We can have eukaryotic cells that form a slime layer. We have eukaryotic cells that form a capsule. I understand this week that you did negative staining, right? So you could outline it. You used either India ink or nigrosin or something like that, right? Okay, you had to have a lot of light going through it. Yeah. What'd you do it with? Dental tartar? No. Okay, you did. Do the dental tartar? Yeah. Very easy. And you saw the little tiny dots in there, bacteria. Yeast do that too. Yeast, which is a eukaryotic cell, have a capsule. So just yeast, the yeast that you're using for bakers or brewers yeast. So all of that's definitely relevant. Protection, adherence, and signal reception, all part of it. So the boundary, and now understand when they get to this, all cells have this, but they do not necessarily have to, it can be have a cell wall or not. Protozoans and helminths, which we don't do not have a cell wall. They're very similar to a typical eukaryotic cell that we would have that functions independently. Like say a white blood cell would be an example of that. Fungi And algae have that cell wall. The algae, because it has chlorophyll, is very much resembling a microscopic plant because it can be single or multicellular. Fungi are the oddball. They have a cell wall like a plant, but they don't have chlorophyll. So that it's almost like it's an animal cell with a cell wall. They're sort of, it's almost a very unique kind of thing that's associated with them because they kind of do business more like we do. Would be the best way to describe it. They're more similar to us in that regard, with the exception of the cell wall, in my opinion. So algae, water-based. Protozoans can be water or soil-based. Fungi, land-based. Fungi and yeast, things like that, mold, land-based. Of course, they get into the air, but that's different because when they're released, they can get into the air. Rigid and structural support, different chemical composition, different kinds of fibers that are there. Fungi, when they're very, very thick and tough, if you ever get a mushroom and you have a stem that's really, really tough, typically it is either cellulose or frequently chitin. Chitin, if you don't know it, is what makes up the exoskeleton of a bug or crab legs, which are basically in the same category. Crustaceans or arthropods. So they have a chitinous exoskeleton. We're having that for the Super Bowl. My wife wants crab legs for the Super Bowl. So much Old Bay, so little time. And so chitin's really important, and all of those things that are there. So we really have to be aware of that. The interesting thing about all of these is their membranes, whether they have a cell wall or not. Plants, they have sterols. You might remember we talked about mycoplasma. Mycoplasma, without a really well-developed even though it's a bacteria, the cell wall doesn't form. It uses sterols to maintain its structural integrity. It has different shapes, but it doesn't explode. Fungi, this is super important. Their sterol, their cholesterol derivative, is called ergosterol. When you get into antifungals, all modern antifungals target ergosterol. If you can't make it, then it can be destroyed. And if you're in a clinical field, I had diabetic patients. Diabetic patients get diabetic female patients get tons of vaginal yeast infections. They're very predisposed. I can't tell you how many diflucan prescriptions I wrote. And fluconazole targets its mode of action, ergosterol synthesis. We'll go over that again when we get to antifungals. And so, I mean, they're demonstrating it here. I don't think this is such a big player. So again, thankfully, the membrane is the membrane. It's phospholipid bilayer. Nothing really overly exciting. Here are the sterols. Relative rigidity and stability to the membrane. And so particularly for like protozoans or for our cells, it's so important. That's why and I probably emphasized the point in A&P, we sort of undervalue the importance of cholesterol. Cholesterol's gotten such a bad rap. Oh my God, and it's really not cholesterol. It has more to do with diet. And if anything, there's a lot of healthy fats. And if your diet is that, along with exercise, a la the keto diet, and you do it properly, you're going to find we have more problems with simple carbohydrates than we do with cholesterol substances. Like years ago, when everybody's, oh, we got bad cholesterol, got to do away with it. They had cookies. They had a whole line of snacks called Snackwells. There's no cholesterol in those. They were all sugar. They couldn't have done anything stupider. Oh, they tasted good, but they were all sugar. They were all carbohydrates. So even they have gone the way, as they say, of the dodo bird. So all of that's important. And again, a nucleus. We know about the nucleus. We know it's got two layers. Okay, it's got an outer and inner layer. And because of that unique kind of a structure in those nuclear pores, remember, large molecules, DNA can't get through there. It's too big. RNA, proteins, nutrients, a variety of things. All the things we need to assemble DNA can come in, but only RNA can go out. That's why transcription, basically changing the message of DNA to RNA, occurs in the nucleus. Translation, changing the message of RNA into proteins, occurs in the cytoplasm. And that's true whether it's a bacteria or a eukaryotic cell, except eukaryotic cells, that's not true. Transcription and translation are the same, but in eukaryotic cells, transcription is in the nucleus, translation is in the cytoplasm. Since effectively it's all in the cytoplasm in prokaryotic cells. And so that's really important. And we went over that. Always a test question. You remember from last time. Don't you dare get it wrong. Where is ribosomal RNA made? It's in the nucleolus. Don't blank that up. I'm studying your test, Maddie, immediately. I'm going back to last year, and I'll see if I have that result. You got it wrong. 20 lashes with a wet noodle. It's my senior English teacher used to use that. So, and chromatin is that, remember, it's strands, and we can't see it. And then chromosomes are when it gets away from those histone proteins. And we'll stop here. I can't do rough and smooth endoplasmic reticulum right now. Have a great weekend. I will try to start working on that dictation and stuff like that.